Good morning and thank you for joining the Vectus Biosystems Limited Investor Webinar. I'm Jane Morgan, the Investor and Media Relations Manager. Today, I am joined by the company's Non-Executive Director and Chairman, Dr. Ronald Schneer, Non-Executive Director and Deputy Chairman, Maury Stang, and Executive Director and Chief Executive Officer, Dr. Karen Duggan. To ask a question throughout the presentation, please use the Q&A function at the bottom of the screen. Maury, I'll now pass over to you.
Thanks very much, Jane, and welcome everybody. Vectus Biosystems is at a very exciting time in its development, being in the clinic, and I'd like to just touch on a few points. So the company itself has a board of directors chaired by Dr. Ronald Schneer, as Jane mentioned. Myself as non-executive director, Karen Duggan as CEO and co-founder, Peter Bush as a CA, and Dr. Susan Pond, who has extensive background in the biotechnology industry. The company has just over 31 million shares on issue and a market capitalization of just over $50 million and a very powerful and broad stream of IP, and I think it's very noteworthy that you'll hear about our patent portfolio, and whilst it's been granted in all key jurisdictions, it has an extensive patent life remaining. The core business is to take an insight into the function of a peptide, which has now been made into a mimetic, a small molecule, which not only seeks to slow down the disease progression, but in fact reverse it. And at this point, let me introduce Ron, and over to you, Ron.
Thanks and welcome, everybody. I just want to premise by saying it's a very exciting company to be involved with because of the potential medical and social impact. I'll just go through some of the highlights where we are today, and then talk a little bit about what we intend to do next year. So as we sit today, we have three in-class assets, and I'll come to a little bit on the next slide on the unmet need of why fibrosis is so important and why to reverse this is even more important. So our VB004 drug is in phase 1, and Karen will talk a lot more about this in her part of the presentation. It addresses systolic hypertension, and none of the other drugs, antihypertensive, really deal with systolic hypertension, and our drug does. And it also can reverse fibrosis in both the heart and lungs. Now, there are other antifibrotics on the market, and none of them have been shown to reverse fibrosis. So we're quite in a unique space, not only because of its efficacy, but there's no other drug at the moment that can actually reverse fibrosis. Drug VB44832, this deals with liver fibrosis and fatty liver of various types, be it due to alcohol or non-alcoholic. And to give you some idea, this is not only a very widespread issue in Western societies, but when you go to the East, there are millions and millions of patients that are potentially affected by hepatitis and other diseases of the liver that could potentially benefit from this drug. And then we have, which is quite topical these days, VBA-79, and that reverses pulmonary fibrosis. I think everybody on the call would understand that fibrosis is one of the complications of COVID, but of a whole host of other pulmonary diseases, from people getting exposed as part of their profession through to post-treatment fibrosis, fibrosis due to medications, etc. So it's another very big and another market that will have great effect. We have a large patent portfolio, and as Maury's already alluded to, we have very strong patent registration. And if you can see, just for our VB004 drug, in all the appropriate jurisdictions, that's North America, Europe, Japan, China, etc., even in Russia and countries like the Philippines and South Africa. We did a capital raise last year, which raised $7 million, and that was to enable us to put in place our phase 1 trial, which Karen will speak to in a moment. And on the board, which is quite a small board, there are people quite experienced in all aspects of biotech and taking this type of product to market. Next slide, please. Just to give you some context on why is fibrosis important, and fibrosis is really the end point of disease and insult to the body, be it trauma, be it due to disease, be it following radiotherapy, be it following infections, etc. So remember, fibrosis is just an endpoint and has huge clinical significance. If you have a heart attack and your heart gets scarred, or if you have insult to your kidney, fibrosis is the endpoint. And so if you look at the 4 organs on the slide, heart failure is a huge item in medical care. And in the US alone, they spend somewhere around $32 billion, and that was in 2013. So if you look at the fact that medical expenses grow at around 10 to 17% a year, that number is going to be much larger today. Liver failure, we've already alluded to, huge number of people in China. So 40% of the population of China has some form of liver disease. Same in India and other areas in Asia where there are large populations. Renal failure, we all know people who have had kidney failure. Dialysis is not an inconvenient, it has complications and is a very expensive treatment. So not only is it better for patients to avoid that, but it's also better for healthcare in terms of cost effectiveness. And if you just look in America alone, in 2011, that's 10 years ago, the cost of kidney failure to the health system was just under $50 billion. And respiratory fibrosis, which causes respiratory failure from mild shortness of breath to death, is another big and growing problem. For example, with pollution and even in mining, black lung is a growing problem, even though there are lots of additional safety measures in place. So next year, what's on our agenda? We'll complete this phase 1 trial. Now, for those that don't know, a phase 1 trial merely looks at the toxicity of a drug, and we're very confident of completing this trial successfully. We have had initial conversations, but we'll certainly reignite our more detailed conversations once we get through phase 1 trial with global pharmaceuticals. If you look historically with these type of drugs, once you get through phase 1 trial, there's great interest and a lot of potential investment from drug companies. And then start looking at other disease entities like COVID specifically. For our A32 drug, that's our liver drug, start undertaking synthesis and do some the initial toxicity studies. We have a whole library of compounds, and already mentioned these two. We're going to look at what drug companies and in fact medical certification boards want to know is look at the mechanisms of how drug works, not only what the clinical outcome is, and we'll put a lot of work into working out the mechanisms as we have, and we're well down the line with VB0004. We need to expand our laboratory and our facilities to expand our drug program because we have a lot of orphan drugs waiting to be developed. And again, just some diseases that might resonate with people on the call. If you look at fibrosis, such as an osteoarthritis, there's a lot of evidence that the pain of osteoarthritis actually now is related not to the bone, but to the fibrosis around joints. We'd like to have a look at that. I think that again has huge morbidity impact in the population. Retinal fibrosis, particularly in undeveloped countries, and Alzheimer's disease. If you look at what happens in the brain, it's the accumulation of these abnormal proteins in the brain are actually the way the brain reacts, and it's the brain form of fibrosis. Now, Accugen, I won't go into the detail of Accugen in terms of its mechanism, but more to show that how agile the science in our company is, because basically this is something that was developed by Karen and her team to allow quantification in PCR experiments, which is simpler, more accurate, more efficient and fast. So the inventiveness of our team to solve problems in science, it's just a very good example. I'll now hand over to Karen.
Thanks, Ron. This is, you'll see a recurring slide, basically designed to take you through the criteria that we find pharma want in new investigational agents. Next slide, please. So the first thing we're looking for is a validated target. Our first drug, VB0004, is an analog taken orally of a naturally occurring peptide called VIP. And my research found that VIP was reduced in the heart, in the failing heart, in line with the amount of fibrosis that was present. More importantly, we've subsequently shown, as you can see in these two papers, that you can reverse existing disease in heart and in kidney with treatment with VIP. So that's the target, and the target's been validated, it's been published. Next slide, please. Next slide. Thank you. The next ones we are looking for are things termed platform technology, i.e., can your drug treat more than one condition? Secondly, is it transformational? By transformational, pharma mean can it reverse existing disease? And then lastly, have you demonstrated that it can in an animal model? Next slide, please. And this is 0004 and systolic blood pressure. And you can see various graphs that are looking at, on the left, the dose of 0004 and the reduction in blood pressure, the maximal dose used and the effect over time. And then on the last one, whether if you stop the drug, you have some sustained effect, but a switch off. And the important things to note are we have a good dose response curve against the top number, the systolic pressure, and it is 3 or 4 times the efficacy of enalapril at similar dose. Enalapril is still widely used and is very much representative of all the best currently available antihypertensive agents. Next slide, please. Next slide. Thank you. And looking at 0004 in the heart, down on the bottom left, you have treatment at 3 doses from right to left, increasing dose. And the open bar, the white bar, is how much fibrosis was present when we started treatment. And in an animal that received no treatment over the 4-week period, on the far right bar, the darkest one, you can see that you had progression of the fibrosis over that 4 weeks, but an intervention with 0004 showed decreasing amounts of fibrosis as dose increased, but most importantly, at the 500 dose, we found that it reversed the fibrosis that was already present. And looking at the representative histology, you can see that at 14 weeks, there's the cyano or blue is the actual fibrotic tissue. The dark red is the heart muscle, and you can see it surrounding blood vessels and heading out between muscle fibers. At 18 weeks, in the control animals, you can see large bands encircling muscle fibers and probably strangling them. But with treatment, you can see there's no fibrosis and we have a normal heart. Next slide, please. In the kidney, a similar story, and we look at a type of fibrosis that puts you onto dialysis. There are 2 parts to fibrosis in the kidney: the filtering units called the glomeruli and the interstitium. And it is interstitial fibrosis that actually decides whether you wind up on dialysis or not. On the lower left, you can see the same sort of dose response curve in the kidney as in the heart, but it's better. At all doses, fibrosis was reversed. And looking at the histology on the interstitium, you look at what are termed tubules. You can see the round structures of cells, and around them are the blue fibrotic bits, and it surrounds parts of some but not all tubules. By 18 weeks, you can see the blue is surrounding most tubules and virtually all the way around all of them. But at 18 weeks with 0004 treatment, there's no fibrosis at all present. Next slide, please. And in the lung, we've used a model using bleomycin, a drug commonly used in cancer chemotherapy. And often people have to have their cancer treatment interrupted because the damage to their lungs is so great that you can no longer continue to treat the cancer. So this is a very commonly used model in animals. We allow 2 weeks for the bleomycin to do its work, so it should be a 16-week control on the top, on the right, and then allow for another 4 weeks, and you can see progression of the amount of fibrosis, which appears as the cyano. The red dots are red blood cells in very fine blood vessels called capillaries. And you can see a loss of these capillaries over that 4-week period and an increase in the fibrosis. So it makes it harder for oxygen to get from the white spaces, the air spaces, across to the red blood cells. But treatment with 0004 down the bottom, you can see has restored all the capillaries and removed all the fibrotic tissue. So you then have the capacity for gas exchange again. Next slide, please. So our next ones are to look at the demonstrated safety in a very large panel of tests conducted in 2 species. This is termed IND toxicology. Next slide, thanks. And we did a large panel, which is summarized on this slide. I won't go through it in detail, but the highlights are really that in a 7-day multiple ascending dose, and that means repeated dose, we gave the highest dose of 2,000 milligrams per kilogram in dogs, in 10-kilogram dogs. So that's 20 grams a day, many, many times more than we will ever give a human. And no adverse events were noted. And at 28 days, we didn't have enough drug to do the 20 grams, so we gave 5 grams a day, and still no adverse events over that period of time. All of the other safety panel measurements were non-events. There were no adverse events. Next slide, please. The next thing is, can we make it? And is the cost of goods competitive? Next slide. 0004 has now been made by 2 companies, so we've demonstrated successful transfer of tech transfer of the synthesis, and it's been made up to a 5-kilogram scale. And at that scale, which is very small compared to what pharma might do, we have a cost of 5 cents per milligram, and that would be expected to go down with increasing scale. So very cost effective and very cost competitive. Our second synthesis has undertaken 3 batches to confirm the consistency of the synthetic process, and our original synthesis showed stability at 2 years, and we have ongoing stability tests on the second synthesis. We would not expect those to be any different from the first synthesis. Next slide, please. The next thing that pharma is looking for is about your IP, and whether it's covering both the compositions of matter and the methods of use. So do we own the drug itself, and as well as its use, and how long will the patent be alive for? Next slide, please. So our patent for 0004 covers the compositions of matter as well as methods of use. The patent life its priority date is September 2014. So we have 13 years, and then on licensing, an additional 5 years. But we do have an extra fillip in our patent portfolio for 0004, and that is the method of synthesis. We have a patent on that, and that's a later patent, trailing another 2 or 3 years behind 0004, and that would give us a further 2-3 years on that patent life. And to validate the patents, they have been granted in all major jurisdictions. Next slide, please. Okay, the next part is the phase 1 trial, which is currently in progress. Next slide, please. Okay, so we engaged Syneos Health as the CRO to write the investigator brochure and the trial protocol and monitor the phase 1. We are doing a conventional single ascending dose, so that's just increasing doses of VB0004 given to cohorts of 8 people, of which 6 are active treatments, 2 are placebo. We are currently at the 3rd stage of that, so we've done 2 milligrams, 10 milligrams, and currently doing 30 milligram dose. There are 2 more doses to go, and then we will undertake what is termed a MAD for 14 days. This is multiple ascending dose. And that means taking the same dose for 14 days, and we will be doing starting that once we get to the 100 milligram dose, and being able to start at lower dose and follow along behind. And then we have a 1B segment to the trial once the 14-day MAD is completed, where we will do 2 groups at 2 different doses in uncomplicated high blood pressure. We've identified the biomarkers, and so we will get pharmacokinetic and pharmacodynamic data out of these studies. So our expected outcomes are a maximum tolerated dose, any dose limiting toxicity. I should say that so far we have seen no adverse events, and the pharmacokinetic and pharmacodynamic data. The second long terminology just means the efficacy of the drug for dose. And as I've said, the first 2 cohorts have been completed. Next slide, please. This just shows you that where the bulk of the deals have been done and their value on a log scale. And you can see that most of the actual pharma deals have been done around the phase 1, either at completion or during phase 1. Very little before and not that many afterwards. So we are in very much the sweet spot of drug development. Next slide, please. Just to take you through the fibrotic liver disease story, for which we have a relative of VB0004, and that's VB4A32. Next slide, please. And this just gives you a list of the causes of cirrhosis, and the big players really are in Asia: infections, Hep B and Hep C. In the Western world, obesity, diabetes, and alcohol, probably in that order. And then a lot of others, also RANs. There are current therapies, vaccination to treat Hep A and Hep B. There's now treatment for Hep C, but interestingly, it doesn't prevent ongoing fibrosis. Although it clears the virus, ongoing damage to the liver occurs. So very much something is needed to complement the treatment of Hep C in the very large Asian market. Next slide, please. There's a large number of potential therapies, and really it could be summed up with mostly ineffective with plus or minus a high incidence of side effects. Next slide, please. And this shows A32, and on the lower left you can see after 6 weeks treatment, either the control animals with quite a bit of fibrosis around blood vessels termed the portal tracts, and it's exuding out between the hepatic cells, which are termed sinusoids, and extending into the sinusoidal spaces. 6 weeks treatment with VB4A32, and you have no fibrosis present and normal, what is termed normal sinusoidal architecture. A graph just shows the dose response curve and the two graphs, the upper one dose response curve and the bottom one the effect on liver function tests, which is improvement. Next slide, please. As has already been alluded to, we have also another drug focusing on lung disease. Not all lung disease it wants a drug that will lower blood pressure as well as treat the fibrosis. So the thing about A79 as opposed to VB0004 is that it doesn't lower blood pressure, so has a potential application to the remainder of the disease categories. Next slide, please. Okay, there's a large number of causes. There are two current therapies, pirfenidone and nintedanib, and the real problem with both of those is a very high incidence of side effects. So they have a high discontinuation rate, or they need to reduce the dose, and both will slow the decline in lung function, but have not actually shown any reversal of pre-existing disease. So a game-changing, there's room for a game-changing agent that could actually reverse disease. Next slide, please. Next slide. And this just shows the effect of A79 on blood pressure, which is none. Next slide, please. And yeah, and this graph you've already seen in the 0004 section, but it does show, confirms that A79 reduces the fibrosis in 4 weeks treatment in Blair-Marson treated rats. Next slide, please. And looking at the histology and the two as previously shown, and the third from the top is the A79, and you can see reinstitution of the capillaries and absolute removal of all the fibrotic cyano colored tissue. Next slide, please. Okay, this just gives you our patent portfolio. So there's a large number of families of patents and many have been granted in various jurisdictions. Next slide, please. This goes on. We believe that we may have a role in the post-COVID syndromes, one of which is fibrotic disease affecting the lungs, the heart, the kidney. There have been lung transplants and kidney transplants as a result of post-COVID fibrosis. Now, we all know the cartoon is basically, we all know the virus looks like a hedgehog, and the little yellow bits that you can see are the ACE2. We've learnt that ACE2 is COVID's SARS-CoV-2 receptor, but it does have a day job, and its day job is to actually break down angiotensin II. So if the virus is sitting on ACE2, not only is the virus getting itself into cells, it is preventing the cells from actually breaking down angiotensin II. Angiotensin II is a bit of a pest if it's around. It can increase its own synthesis by increasing its precursor, angiotensinogen, which will in turn go to increased angiotensin II, which increases TNF-alpha, and TNF-alpha will increase the receptor for angiotensin II, the AT1A receptor. All of that is a cycle of increasing fibrosis. Meanwhile, the thing that we're most concerned about at the moment is the angiotensin II will trigger the cytokine storm and the acute respiratory distress syndrome. So ventilation, which is another road to fibrosis. Next slide, please. So this is just showing that both 0004, which has been published, reduces the precursor angiotensinogen, the AT1A receptor expression, and TNF-alpha receptor expression, and as does our drug. So we believe that we will also have a role in the post-COVID fibrosis because we're interrupting the mechanism that is producing fibrosis following this disease. Next slide, please. And another area that we think we have a role in is, and have started work on, is Alzheimer's disease. It's a protein accumulation disease in the brain. The protein is amyloid rather than collagen and fibronectin, but it does the same thing. It stops the neurons, in this case, communicating with one another and acting effectively. It causes their atrophy and death, and essentially it's a fibrosis of the brain. And as you can see in the macroscopic or large-scale histology, severe Alzheimer's, you get a shrinking of the brain in comparison to the normal brain. Again, one of our agents, we believe, if we can find the one that will cross the blood-brain barrier the best, will be able to get in there, cause that protein to be removed, and hopefully regeneration of the neurons in the brain and reversal of the Alzheimer's.
So thanks, Karen. Before we open up for some questions, I just want to recap at a very high level. We all know that biotechnology is a very, very lucrative and valuable outcome for a successful company. But I think we need to make a strong point of how much Vectus has de-risked this, this journey. We have the IP portfolio. We have a platform technology. We have granted patents. We have patents that cover discovery, utility, and mode of manufacture. We have clinical trials in humans now, which are beginning to reflect the data we saw preclinically. And importantly, because the entities that Vectus has are very, very novel, it's very likely that we're going to be looking at first-in-class reimbursement. And that has phenomenal effect in the value of the company. And globally, this represents some of the largest disease states in the world. So we're broad, we're deep, and we are advanced on every front. When we look at the toxicity trials, when we look at the cost of manufacture, we look at the pharmaceutical scale-up, we look at the fact that the ATO is now approved for us to do work not only in Australia to get the cash back, but overseas as well. And we're regarded by the broad cross-section of the pharmaceutical industry as being a credible and very, very exciting emerging company. So at that point, I'll open up to some questions. Jane, over to you.
Wonderful, thank you. And I do encourage webinar attendees to use the Q&A function at the bottom of the screen, but I will jump into them, and a lot of them were answered throughout the presentation, but I'll just in case anyone was missed the presentation. So could the compounds aid with the treatment of COVID vaccine related injuries?
We haven't, I guess the vaccine related injuries, one would have the myocarditis with Pfizer and possibly occurs with Moderna is self-limited and appears not to have any long-term sequelae. The AstraZeneca TTS, there does seem to be some infarction. It's very difficult to know. There's not a lot of data there, possibly, depending on whether fibrotic damage then occurs from those side effects.
Another one's come through. So what is the route of administration for 0004 and A32?
They're oral pills that you would just take like any other medication.
Jane, that's really an excellent point. We have pointed out to the market that as part of our success, we have a technology that allows us to take what are conventional peptide therapeutics and turn them into small molecules that are orally dosable. This is a huge and important facet of our know-how. And rather than self-injecting on a daily basis, we're projecting one pill a day, and those pills will be reimbursed on a health economic basis. So what do I mean by that? We're not a generic. And therefore, in the case of a dialysis patient that can cost $50,000 to $150,000 a year, in addition to the social impacts that Ron spoke about, we can be reimbursed literally on the savings we give to managing that patient. Now, obviously, our interest is the patient never to go on to dialysis. So we're in a very, very unique and powerful position.
Thank you for that. There's been a few questions actually coming through about a big pharma deal and what that would sort of look like and what the timing's like for that. Maury, I might hand to you for that one.
Yes, so as we've outlined to the market prior to COVID, when we could travel, we have had interactions and very constructive interactions with quite a large number of pharmaceutical companies. Karen and I have attended the San Diego global meetings and the San Francisco JP Morgan meetings. And so typically we are saying today that we're undertaking a process of re-engaging with pharma around the world. It would involve a upfront payment. If we look at a company like FIBROTEC, they had a package of about $600 million for an existing compound that they repurposed. It would also involve milestone payments. And then finally, we could expect first-in-class reimbursement at the higher end of the range. And pharmaceutical range ranges from 12 to maybe 28% as a royalty payment. And even at the midpoint there, and a multi-billion-dollar drug, you have a very, very good outcome for our shareholders.
Wonderful. There's one that's come through just on, so can you remind us on how phase 1 is progressing and the expected completion date for 1A and 1B?
We're probably about a third to halfway through phase 1. The SAD of phase 1 and our recruitment has been slowed a bit by the severe lockdown that occurred in Melbourne. That now appears to be picking up. Our completion date will be Q1, late Q1 next year for the 1A, and Q2, perhaps early Q3 for the phase 1B.
Wonderful. That looks like all the questions have come through. So thank you all for joining us today. And if you do have any other further questions, I encourage you to reach out via the contact details on the bottom of our ASX releases. And a copy of today's recording will be available online in the coming days. Thank you all for joining us.
Thanks,
Thanks, Jane.
everybody. Thanks.